Can hear me okay?
I can hear you fine.
Good. So Sharon, thank you for the invitation. I'd like to thank everybody for joining our discussion tonight, which is certainly a timely topic. So as I described in the title, I'm going to be giving this presentation in 3 segments. First, talk about the virus and the disease, then vaccine development, and then how a vaccine is used in the field. And after each segment, which will last around 15 plus minutes, I'll pause for a few minutes to take questions and answers on that particular segment. So why start with the virus and disease? Well, it's the case with every vaccine that has been developed. And in fact, really every pharmaceutical product that's been developed is that before developing the product, you need to understand the disease. And of course, in this case, the disease and the virus are interwoven. So we need to really understand those. And then that teaches how to best make the vaccine. The virus we're dealing with is a brilliant virus. It's without question the most notorious, most successful virus since the 1918 pandemic flu about 100 years ago. You know the effects it has caused. I don't need to run through the statistics, although if you think about pandemic flu, which killed somewhere between 20 and 50 million people, that was actually about 100-fold more deadly than COVID. If you can imagine that up to hundreds of 100,000, 200,000 people in Israel would die of the disease. That's what happened with pandemic flu. But we learned a lot about pandemic disease at that time, and now some of those lessons are coming true today. The most important thing to realize about the disease itself as it develops is this initial virus infection, and then there's a race between the virus and the immune system to see who will get the upper hand, literally. The immune system starts to recognize the virus within a day or two, and then starts to mount a non-specific attack, then a specific attack with antibodies and immune cells, and meanwhile the virus is replicating. So when there's a heavy load of virus, the virus can get the upper hand and disease results, severe and lethal, depending how advanced it gets. If the virus load is not so high and the individual has a good immune system, then the immune system usually gains the upper hand, because indeed the great majority of infections do not lead to disease. In fact, most don't even lead to symptoms. So in most cases, the immune system has the upper hand, but we see the consequences when the virus gets the upper hand. So as we know, we get antibodies, which will be specific for the virus. And I'm going to mention the term neutralizing antibody. These are antibodies that will neutralize or stop the infectivity of the virus. Those are the key antibodies to protection. And then we have immune cells called T cells, which help also in the immune attack against the virus and also provide immune memory. So that hopefully, if someone encounters the virus now and they recover, and then in a year they encounter the virus again, the memory of that virus will bring back an immune response faster, and the individual is less likely to get ill a second time around. Now, if the virus wins, it's not just a question of the virus replicating, but the virus basically hijacks the body's immune system, and instead of giving protective responses, the body's immune system can result in destructive responses. And you've heard these terms like cytokine storm and cytokine damage. These are basically inflammatory type responses from your immune system. And there also may be, in some cases, autoimmune type responses where your immune system reacts against body components, and that itself causes disease as well. So that's the race battle that takes place early after infection. Now, other things to learn about the virus, this is actually the 7th known coronavirus that will infect people. 4 of these are causes of the common cold. And in fact, coronavirus is the 2nd most common cause of the common cold, the leading one being rhinovirus. And then there are 2 others which are very notorious viruses, the SARS virus of 2002 and the MERS virus of 2011. These were very highly fatal infections. SARS killed about 10+% of people who were infected. MERS, 30 to 40% of those who were infected, very highly fatal. The difference being those viruses transmitted very poorly from one person to another. So people were acquiring infections from animals, not so much from person to person, and the virus and the disease petered out relatively rapidly. SARS affected thousands of people, MERS maybe hundreds. The difference, as you all know, is that the current virus, current COVID-19 virus, spreads very rapidly and very efficiently. And the work on SARS explained a lot about that. We learned a lot about COVID from SARS in 2002 and everything that was done after that. We learned about a cellular receptor, which is present on most cells in the body. And that's the receptor to which the virus binds to initiate infection. The difference between SARS and COVID is that COVID binds to the same receptor as SARS, but binds much, much more tightly. And that is accounting for its greater infectivity and greater spread than SARS. The other thing we learned from SARS, which was very valuable, is you've seen these pictures of the SARS virus. It looks like a crown. There are these tips that are really beautiful. And it's a beautiful image under the microscope. And from that particular image, we see that there's a protein called the spike protein, which is what you see in these particular virus spikes that come from the surface. And the spike protein, or S protein, as I'll refer to, is the leading vaccine component for SARS vaccine and is now the leading component for COVID, because what it was in SARS is a very related protein that's in COVID. In addition, at the very tip of the spike is the area that it will bind to the cell receptor, and that's called the receptor binding domain, and antibodies against that tip are the key antibodies that will neutralize infection. So again, we learned all of that from SARS, and when COVID came along and the sequence was published, people saw they could rapidly take the lessons learned from SARS and plug them into COVID. There are also animal models of infection for SARS, and that taught about animal models for COVID. And very importantly, we learned that neutralizing antibody was apparently necessary and sufficient to neutralize SARS infection, and that appears to be the case also for COVID infection. And so we learn that's the antibody we want, and we learn also that if we want to test vaccines, we design an assay that will detect the neutralizing antibody. Again, another great lesson from SARS. We also learned about T cells, and the T cells are induced and the T cells assist in protection. We also learned something very notorious about SARS, is that the SARS infection and MERS also could induce the wrong kind of antibody. This is an enhancing antibody. It's an antibody that actually helps the virus to infect more efficiently. So it's a nasty antibody. If you induce that antibody, as was shown in some animal trials, you make the disease worse, not better. So understanding that your vaccine does not induce this bad antibody has been very important for development, and making antibody tests in the lab to detect this enhancing or bad antibody are also very important. Another fascinating observation that came out that speaks to the importance of the T cells, if you look at individuals who have never seen COVID, you'll find that some of them have T cells that will react with COVID. So they have no antibody, no evidence of exposure to COVID, but they have antibodies that react. And apparently what's the case is they have an immune memory of the coronavirus that causes the common cold, which has some reactivity against COVID. And it might be the case that those who have less severe disease or no disease, they may be individuals who have some cross-reactive T cells that gave them this partial protection. And finally, the other very important thing learned from SARS and from MERS is that the antibody that is induced, particularly the neutralizing antibody, is a very short-lived antibody. Its half-life is much less than many other antibodies. Now, you can look at other vaccines, and there are many vaccines that will induce antibody responses that will persist for months, sometimes for years, sometimes a decade or more. It's an antibody that the cells in your blood love to make and doesn't cause any consequence for the body, whereas this antibody is very short-lived. Now, it might be, at least this is a possible explanation, because this antibody binds to the part of the virus that binds to the cell, there are networks of antibodies made in your body, of antibodies and antibodies against antibodies. So an antibody against the neutralizing COVID antibody would actually bind to your cells because it's a mirror image. And by binding to your cells would be deleterious to your body. So your body seems to find a way to tune down the protective antibody, because otherwise the protective antibody might give rise to an antibody that binds to cells, which is bad for the body. Whatever that's one likely explanation, but whatever the explanation, the fact that the antibody is short-lived in SARS and MERS is a very important lesson learned for SARS. And it's been shown for COVID, it's been shown for COVID that subjects who have had disease, that after a few months, their antibodies levels are already dropping significantly. So again, another important lesson learned. So let me recap some of the important points, and then I'm going to stop for a couple of minutes to take some questions. So we've learned how with all virus infections, but particularly this one, there's a battle between the virus and the body's immune system for control. Neutralizing antibody that will stop the virus from binding to cells is the key to infection, just to stopping infection. And assays for the virus neutralizing antibody are key to following the disease and following vaccine development. If the virus gets the upper hand, the virus can turn the immune system against the body. And that is what causes so many of the side effects that we've all read about with COVID. We also know that COVID is one of 7 members of the corona family, and there have been many lessons learned from the other members, particularly about the receptor, the value of neutralizing antibody, the value or negative value of the wrong kind of antibody, the bad antibody, and cells that will cross-react from previous infection, and most importantly, that the antibody is short-lived. So those are the issues that everyone learned about, that scientists learned about in January and February, and that became the basis for then proceeding with vaccine development after that. My next segment will be vaccine development, but I'm going to stop here and take some questions on this particular segment. So anyone who wants to ask a question, only when you're ready to ask, please unmute, and happy to have your questions.
Chaim, did you have one? Chaim Leifman?
Hello?
Yeah, go ahead, Marion.
Ronald, are you going to be talking about herd immunity?
Yes, I will come to that actually in a later segment, the third segment.
Thank you very much. Thank you.
Ronald, there's a question on chat. Are there drugs which can be used to stimulate production of the correct antibodies?
No, there aren't. Drugs generally will not induce an antibody response. Only the vaccine can do that, and only if the vaccine is designed in a way that it will induce the correct antibody, the neutralizing antibody, and not the bad antibody. But drugs will not help with that. Now, there can be other aspects of health that will affect the antibody production. For instance, the general state of the immune system. If somebody has an immune deficiency, they're not going to be able to respond well to the virus. If someone is in good health and has a good immune system, they will generally be able to respond better. Other modulations include the fact that it's known that people who are under stress or depression or anxiety will generally make less antibody and have lower immune responses. So having a good mental state of mind for all antibodies, but particularly antibodies against infectious agents, is important for countering the virus and also important for raising a good immune response. So anybody who's thinking of getting vaccinated, you're bound to be more successfully vaccinated if you're in a positive state of mind than if you're under stress. So that's another issue that will affect the immune response.
Okay.
Is there any other questions on the immune system, on lessons learned, on general properties of the infection from an immunology point of view.
Is there any way to produce a vaccine that will give us a long-term antibody response, as opposed what you say now is all we're getting is a short-term antibody response?
Well, I'd say this, is that there's no way to design it a priori. There are certain vaccine strategies which might increase the likelihood of longer-term production, but that's only theoretical. And the answer to your question will only come in next 3, 4, or 5 years in terms of some of the vaccine platforms, which maybe will allow the ability to give a longer-lived antibody. The problem is not the induction of the antibody per se. There seems to be a problem that the antibody by itself just doesn't seem to persist well. We'll see from the ongoing clinical trials if the persistence of the vaccine-induced antibody is similar to the persistence of the infection-induced antibody. As seems to be the case with other viruses, it's usually, but not always the case, that the vaccine-induced antibody and the natural antibody may have equal long or equally short lives. So it's more, seems to be a property of the antibody and the place on the virus the antibody binds to. So people are trying to do it in ways that may induce longer lived antibody, but it's not at all assured, and we'll have to see how the vaccine trials show the duration of the antibody.
Ron, there are 2 other questions came up on the chat. One is, how does diet work into this? And is someone with an autoimmune disease...
I'm sorry, the first question again, I missed a word you said.
Yeah, how does diet work into this? And is someone with an autoimmune disease more susceptible to COVID? And if he is being treated with Plaquenil, is he less susceptible?
Yeah, I'd say, in terms of the question of health and question of diet, I'd say to the extent to which diet enables someone to have a generally healthy body, which also means a healthy immune system, probably good enough. There are no particular dietary supplements which by themselves may be immune enforcers. There are things that are sold in health food stores that are reputed to be able to help immune responses, but that's not necessarily the case. There are drugs that will enhance immune responses. They'll enhance immune performance in general, but those are very expensive. They're generally not given on a routine basis. In terms of autoimmunity, as I understand, the people who have any kind of immune defect may be more vulnerable to COVID and may respond less well to a vaccine. I don't know the answer to the question of whether someone who is autoimmune and on a particular drug, how they will fare in terms of the disease or a vaccine. I don't know the answer to that. But yes, generally immune deficiencies, autoimmunity, immune defects of different kinds, an individual will be generally more vulnerable to COVID disease and generally respond less well to a vaccine. I say generally, not always.
Yeah, there seems to be one question: what interest does the virus have in killing the host?
I'll take this, by the way, as the last question. There's some viruses, you read about Ebola and some of these other viruses in Africa that can kill 80% of people. They're not very successful. They kill a small cohort of people and then they disappear. A virus does not want to generally kill the host, but on the other hand, if a virus is very efficient in spreading, like this particular virus, if some percent of people die as a result, it doesn't affect the virus that much. The virus may kill 0.3 to 0.5%. And I say that because there are many more people who are infected and don't know it. If you look at the published numbers, about 5 to 10-fold more than that are actually infected. So the mortality rate is maybe 0.3, 0.5%, which means well over 99% of people are not killed. If you're a virus, that's actually pretty good. And the virus, as you see, spreads efficiently. It's one of the best spreaders of any virus in any virus kingdom. Maybe I'm going to move on now to vaccine development and take that as a lead-in. And I want to start with one interesting question which comes up. I mean, we see where we are now in phase 3 trials, less than 1 year after the virus was discovered. Usually it takes vaccines 10 years from the discovery or beginning of vaccine development until they're ready for phase 3 trial, 15 years to hit the market. And this is much faster. I mean, it is mind-boggling how much faster it is. I want to, again, reference back to some of the issues I discussed before, that there's a great deal of knowledge from SARS. We learned about the proteins. We learned about neutralizing antibodies, and this became a basis that enabled companies to jump in very quickly and make vaccines that worked well in the laboratory and in animal models and get to the clinic quickly and have tests to show that people make neutralizing antibody. The other big issue is that companies develop vaccines for a long time. They have to justify the investment. They have to find investment. The investments compete with other projects in the same company, and they need to earn a good enough rate of return or the company will not work on it. Well, in this case, obviously, the investment is well justified. The political and ethical mandate is extremely high. And there's, I won't say a bottomless pit of money, but the money available from the US government is many billions. There are donor organizations, including Gates Foundation, that is giving $2 billion or more for manufacturing. And because there's so much money available, along with the great knowledge base and the great ethical political mandate, it's all made things go extremely quickly. And different companies have gotten in, and what companies have done is they've worked on the vaccine platform, a way of making a vaccine in which they have had successes in other projects. So because they had an available platform, a way to make other vaccines, to plug in the S protein, which is the vaccine antigen, and get it to work in their platform was relatively straightforward and relatively quick. And you can read about these biotech companies that said they got the sequence and a week later they had a vaccine made. Well, it's actually true. They actually had a system all ready to go because they could simply plug in this new sequence into the vaccine machinery that they already had. And there are different ways to make it. I'm just going to summarize them in a very general way. One is to make and purify the S protein and inject the S protein all by itself. Another is to take cells in culture that are infected with the virus, inactivate them chemically so you kill the virus, and then administer that as well. And there are many cases of vaccines that have worked along this principle. The purified proteins, you have human papilloma vaccine, hepatitis B vaccine, extremely successful, highly effective vaccines. These inactivated vaccines, things like hepatitis A, polio, there are many examples of that. We have these inactivated vaccines which also work very effectively. You have new generation vaccines now, which include RNA and DNA. So you're actually taking the genetic material that encodes the S protein and you're injecting the genetic material, which gets taken up by the cells into whom you inject it, and the cells then manufacture the S protein. So the body is now manufacturing the vaccine all by itself. And then you have the other very clever approach that different companies have used, which is called the virus vector. You take another virus, and I'll mention it as an example, adenovirus, which also causes the common cold. You now take an adenovirus, which is weakened, that cannot cause any disease. You put the sequence of the S protein into this virus, and the virus then infects cells, and these infected cells, infected by this virus, then manufacture S protein. And again, the vaccine is made inside the body, inside the host. Each of these approaches is distinct. They will give different levels of antibody, of the good neutralizing antibody, or of T cells, which help the immune response, some higher, some lower. And we're going to learn over time in all the ongoing trials which ones are giving the highest level of antibodies. And back to the question asked before, whether some of the antibodies may last longer when given by, let's say, DNA than when given by injecting just a protein. We'll also see whether any approaches run the risk of making this bad antibody, and that, of course, would not be a good approach. So all these things are being learned from ongoing trials, and by probably, I'll say next spring, we'll have a very good idea of how all of these different platforms are performing and which ones are potentially giving the best protection against disease. Now, another key for all this development is a standard neutralizing antibody assay, which can be made automated. And if you think about it, you're going to have these clinical trials in 10,000, 30,000 people, and you're going to be assaying and testing for antibodies in huge numbers. Companies manufacture kits in which you can then test very large numbers of blood samples. You bleed somebody after they're injected by the vaccine. You wait a few weeks, you bleed them, you separate the serum from the cells, and then you test the serum in this standard antibody assay and see the level of neutralizing antibody. There are also standard assays now for the bad antibody to see if you're giving an antibody that will enhance protection. And through this, there'll be some knowledge of how many doses you give and what the schedule for injection is if you're giving more than one dose. And a great standardization will occur. One of the other important tests will be to test the vaccine-induced antibodies compared to natural antibodies. If you have a subject that has been infected with COVID, then this particular subject will have neutralizing antibodies after they recover. And you look to see the level of neutralizing antibodies in someone who has recovered from infection. Then you ask the question, does the vaccine induce a level of antibodies at least as high or higher than you see in these sera? And you might take 20 or 30, a panel of sera, because the human response is diverse, of people who have recovered, and see if the vaccine will induce about the same or higher level. And you'll see reports in the press about how vaccine companies say they can induce as much or more antibody than you find in people who are recovered. Very, very important. If you don't make as much antibody as in someone recovered, then your vaccine might not be good enough. So that's considered informally an important hurdle to surpass. Now, I know we talked about what affects the immune response. I talked about health, immune state, stress. Age is a determinant because age, in general, with vaccines, the older an individual is, the less well they respond. And you can track immune responses decade by decade through life, as been done with, say, hepatitis B. And you can show that decade by decade, the average immune response is less and less. And yet with COVID, of course, the key is to induce immune response in older subjects being the ones who are most vulnerable. So testing in different groups is important. Testing in people of high risk, you want to test in people who, for instance, have certain underlying medical conditions. And it might be that with certain vaccines, you need to give an additional dose or a higher dose to people who don't respond as well. So you track the immune response in order to learn about how to administer the vaccine. And now through all of this, you'll have the profile of each individual vaccine. It will not really be possible to compare vaccine A and vaccine B. Each of them makes a claim of how well it performs, but each of them is tested on its own, tested in different assays, and not tested head-to-head. So each of them will be tested in an efficacy trial, and when they're in an efficacy trial, then we will see which ones protect better. We'll also learn about antibody persistence. Does the antibody last an average half-life of 2 weeks or 4 weeks or more? Is it like the natural antibody, or does it last longer? Furthermore, in the assumption that the vaccine will induce an antibody that's not long-lived, when do you need to give a booster down the road? It might be, and I'd say from what we know today, it might be every year, if you've been vaccinated with COVID, every year you get boosted because the antibody response and the immune response doesn't last long enough. Another very important issue is going to be how well tolerated the vaccine is. That is, are there injection site complaints or other things that happen close after injection, or safety issues. In other words, you get other side effects that may show up later after injection, and safety has to be looked at very closely. Now we get to some of the big and difficult questions. So how effective is effective? What is the hurdle that a vaccine has to pass in order to be licensed for use? What if it's 30% effective or 50% or 70%? 70% meaning that a subject has a 70% chance of being protected if they are injected with the vaccine, or 50% likewise. To get reliable numbers, you have to do very large scale trials, and you see these trials are in the tens of thousands. If you have only a few people and you're testing to see how effectively the vaccine works, you're not going to get a very accurate number. So companies go into very large numbers in order to get an accurate readout on how effective the vaccine is. The early vaccines might be less effective than vaccines that are developed in a year or two. And as people work more on vaccines and take lessons learned from the first generation vaccines, there may be better vaccines. Or they might learn that an efficacy trial giving an additional dose would give more protection. So the initial efficacy results might not be as high as efficacy results generated in 2 or 3 years. People want the vaccine quickly, but if you release a vaccine that has too low a rate of efficacy, people may think to themselves, now I'm protected, I don't need a mask, I don't need a social distance, and they're vulnerable to infection. If you wait too long for a vaccine that's very highly efficacious, like 80 or 90%, then people are not benefiting from a vaccine and the virus is still continuing to circulate. So you need some middle ground between those, and there are debates already ongoing on that. Then you have the issue of safety. How safe is safe? I'm not talking about, let's say you inject flu vaccine, your arm turns a little red. That may happen with existing vaccines, or you get a slight fever. But I'm talking about some serious side effects. And like every medical product, there are vaccines that can have serious side effects. And most vaccines on the market do not give very serious side effects. And I mean by serious side effects, effects on an organ, effects on the heart, permanent damage of some kind, usually less than 1 in 100,000, less serious side effects, but still noticeable side effects, generally less than 1 in 10,000. And people have focused on the number of 1 in 10,000 as maybe being a level that is a reasonable compromise between being safe enough and not having to be so safe that you'd have to test hundreds of thousands of people to show that it's ultra safe. Again, it's an ongoing debate. So let's say you want a vaccine that gives that doesn't have more than 1 in 10,000 serious side effects. If you go through a statistical calculation, if you want to look at for 1 in 10,000, if you immunize 30,000 people, there is a 95% likelihood that you can detect something as uncommon as 1 in 10,000. If you need to go for 1 in 100,000, you'd have to immunize 300,000 people, which would take years in order to detect something reliably of 1 in 100,000. So again, people focus on this 1 in 10,000. That's why you're seeing these clinical trials in the range of tens of thousands of individuals. There are other ways of looking at safety. For instance, you might release a vaccine for use. That's after being tested in tens of thousands of people, and then you'll look for side effects after the vaccine is licensed. So there's a system in the United States that tracks all side effects on all licensed vaccines. It's done by the US CDC. They've collected maybe 30,000 complaints a year from different vaccines, and they look for patterns in these complaints. In some of the worst cases where there was a significant number of cases, there was a vaccine for diarrhea in infants that was released about 20 years ago that caused significant side effects that were so significant the vaccine was withdrawn. There are other cases where there may be changes in the label of the vaccine to warn people that if you take the vaccine that at a very low rate, you might get some other side effect, whatever it may be, let's say a stomach-related side effect. And in that way, through this kind of surveillance after the vaccine's on the market, you can track safety. So not just what you see in clinical trials, but what you see after clinical trials. Now, going too quickly has a problem, and there have been certain other vaccines that have been developed in an emergency situation. There is a pandemic flu vaccine developed in the 2000s, a swine flu vaccine in the 70s that was developed very quickly. The swine flu became notorious because it gave rise to an association with Guillain-Barré syndrome, which is a facial paralysis. And again, it was done in a way that was almost like an emergency, and people went so quickly, and public health officials got behind it so quickly that not enough care was taken in the overall program. So we don't want to do that either. There needs to be a balance between going fast enough to benefit the population, but slow enough to be prudent. And this is, again, part of this entire ongoing debate to make sure that we're balancing safety and effectiveness, benefits to the population and safety to the population. As a background, further background to all this, we also have manufacturing. Now take into account that a vaccine that is made in a lab needs to be scaled up perhaps 10,000-fold or more in a manufacturing facility. And you've seen how many companies are already starting to manufacture vaccine. They're being paid actually by the US government, who gives a grant to manufacture, because otherwise a company may invest $1 billion in manufacturing a vaccine and then the vaccine doesn't work in the phase 3 trial. So US government and Gates are underwriting a lot of these costs. A facility has to be renovated. You need manufacturing equipment specific for how you manufacture your vaccine. If it's a purified protein or an RNA or DNA or an inactive vaccine, the equipment and the way in which you manufacture may be totally different. In addition, you need manufacturing capability after you make the vaccine. You need to be able to fill the vaccine into vials or syringes. You need packaging facilities. If you're going to be releasing tens of millions of doses, you need automated lines to put vials into packages and make packages in a very convenient way. You need supply of hundreds of millions of vials into which you're going to fill. So all of this is taking place at the same time. Now, we're talking about a vaccine that arguably we may need billions of doses. It's mind-boggling that to think in a year or two we might need billions to vaccinate most of the world. Keep in mind that for current vaccine manufacturing facilities for routinely used vaccines, a vaccine facility making 100 million doses a year is considered a large facility. 100 million is not going to do here. If you want to manufacture 2 or 3 billion doses, you can do the math. You need something much larger. So companies are already trying to make these very large facilities or duplicate facilities to make this huge amount of vaccine as quickly as possible. So that's all happening simultaneously with all the clinical trials. It's quiet, but it's happening. And there are hundreds, maybe up to 1,000 or more production workers who are being trained, in some cases from the beginning, on how to run manufacturing processes, how to run assays, how to keep a high quality manufacturing operation. The air supply has to be very high quality because you are going to be filling these vaccines in a very special way. So you need very high quality air in which there are very few particles. So you need these very expensive HVAC, these filtration systems to make this very high quality of air. You need facilities where you make very, very high quality of water, double distilled, deionized, filtered, so that there's no chance of any other contamination coming in through your water. So that's the other side of things, which is a multi-billion-dollar operation by multiple companies. So it's actually, again, I want to stress the fact, to me, having been in the field for quite a while, it's mind-boggling to me to see everything that's happened in such a short period of time, of having vaccines in the clinic 4 or 5 months after the virus was discovered and sequenced made available, to be able to start clinical trials and within 6 or 7 months be running and enrolling tens of thousands of people, to put billions into production, into facilities that will make hundreds of millions of doses, to have assays available and kits available to test for antibodies, and the big knowledge base that has developed already. So it's a tribute to the field that this has happened so quickly, but it's because there were other coronaviruses, a lot learned from that, and a lot of know-how in the vaccine industry that has made dozens of vaccines that are now on the market, preventing millions of deaths per year. So that's an overview of vaccines, and I'm going to stop here and take any questions that you have on vaccines.
Ron, there is a question on the chat. Is it possible to produce a vaccine in a lab without producing antibodies from the virus?
I'm not sure I understand the question. To make it in the lab, I mean, you're making the vaccine by itself is not the antibody. And the antibody is made after injection into the human host. If you're asking, can you make a vaccine that will only make immune cells and not antibodies? It's very, very difficult to do so. You might make antibodies that are not relevant to neutralization and stimulate the production of immune cells. The only problem there is that it is not known that immune cells by themselves can protect against disease. It is known that antibody will protect, and it seems that antibody is necessary and sufficient. There's no evidence that immune cells are necessary and sufficient, so inducing the antibody seems to be very important. I hope I've answered the question.
Well,
other questions?
somebody wanted you to define an assay, what an assay is.
An assay is a test. So I'll give an example of a test. An individual gets symptoms of COVID, and then you go to one of these testing centers. You go to one of these testing centers, and in the testing center, they will evaluate you. It's actually an assay rather than a test. So an assay is really the kind of scientific term for a test. And the assay is it's usually a kit of some kind. A laboratory can design the kit if it wants, but typically it's made by a manufacturer like Johnson & Johnson or Abbott. And in doing that, they'll have different reagents in the kit. So the kit may have a plastic template with wells in it, and you put reagents into those wells, and then you get a color readout, and that tells you a result. And tests like that are done to show that somebody is infected or not infected. And tests can be done likewise for testing for antibodies in this very automated way. So that's what the assay is. It's basically a test. There is the test has what's called an analyte. It's the thing you're assaying for. So you're analyzing for the virus to see if someone's infected. You're assaying or testing for the antibody when you're looking for the immune response. So that's what the assay accomplishes. Some additional questions on vaccines, how it's made in the lab, production, clinical trials, safety issues, how you look at safety, how you worry about safety. No other questions?
You have a whole bunch of written questions.
Ron, there are a lot of chats. You have to open your chats.
Okay, can somebody say it verbally to me? I don't see the...
When do you think we'll have the vaccine available here?
Tony, I'm not in a non-responsive mode right now.
Say that question. When will the vaccine be available? I'll come to that in the third part. My very short answer is I have no idea. It's unknown.
What do you think about the Russian?
I'm looking at the chat. What do I think of the Russian vaccine? I don't want to get into politics, but it's hard for me to believe that any vaccine that was made and tested and released after being tested for 2 months, that they can really get a good gauge on how safe and effective it is. So I don't believe in any vaccine, whether it's Russian, American, whatever it is, that is tested for 2 months and then released to the population. Doesn't make sense to me that that can be safe or effective. The major vaccine producers include many large pharma companies: Merck, GlaxoSmithKline, Pfizer, AstraZeneca, Sanofi Pasteur, Those are some of the major ones. You read about them all the time in the press. They're also contract manufacturers, simply to do things on contract. How do you make a vaccine to a rapidly mutating virus? Well, the coronavirus throws off a lot of mutations, many more mutations than flu does. To date, none of the mutations has been found to affect the ability of the vaccine to be neutralized by antibodies. So all of the mutations are happening outside of the neutralizing domain, and that's important. And you think of it from this point of view, the neutralizing domain is the cell binding domain. If the neutralizing domain changes in a way to escape neutralization, the virus is not going to bind as well to cells. So the mutations of the virus to get away from antibody will also weaken the virus. So that seems to be the reason that the virus mutation is not a factor, at least what's been observed so far. And there are companies that have looked at tens of thousands of sequences and doing that in a way where they can show that there's no mutation in the key domain. The question about Trump, I'm going to decline to discuss any political questions. I'm going to stick to science and medicine tonight. Those are all the ones I can see. Are there any other questions available on the vaccine? As I said, I'm going to get to the next stage, which is the vaccine being released for use. How and when and by what criteria, who uses it, will people be willing to take it? So are there any other chat questions that I've missed beyond these? Okay, so I guess I've hit everything. So let me then continue, and I'm going to go on to how a vaccine is used in the field. And there have been questions around this, and it's everybody's first question: when will the vaccine be available? And when I give the answer, I have no idea. I'm really answering for the field as a whole. Nobody really knows. There are a lot of things that many people talk about, whether they are public health experts or politicians or physicians or vaccine companies or stock analysts, and people will say all kinds of dates. We can't believe any of those. Here's what needs to happen. The vaccine trial to show efficacy has to be done, and they're all being done now in areas where there is still disease. It's kind of the good news, bad news things. The bad news is the disease is still around, and I'm not saying that that's a good news, but what it has done for vaccine development is enable the field to determine relatively quickly whether the vaccine is preventing disease. You compare the rate of disease in the vaccine group versus the rate that's getting either another vaccine or getting a placebo of some kind, and you can then calculate the percent of disease being prevented. Those trials have to enroll large numbers of people, and it's not known when an enrollment will finish until the company announces it. They will then follow these people for some number of months, and then at the end, they will get all the results and analyze all the results and reveal the degree of safety. Now, that's only part of it. They have to analyze all the safety data. And by safety data, I mean people are then tested for temperature, complete blood workup, complete blood chemistry, blood cell counts have to be taken, physiological functions of all kinds are taken. And it may be, if you have 30,000 people in a trial and you're taking these large numbers of safety parameters, particularly right after injection, however you're doing it, you may have hundreds of thousands, 1 million or more data points. These all have to be analyzed. And particularly if you're going to look at safety-related issues, you're looking for complaints, like fever or injection site complaint. The only way to do it is you're going to compare the vaccine group to the placebo control group. And then from that, you will see what complaints you're getting that relate to the vaccine. Likewise, if you're looking for these very rare side effects, you're going to compare the vaccine group to the placebo group, but you need to then analyze these hundreds of thousands or 1 million or more data points in order to get an accurate readout for efficacy. So they may, for safety, companies may announce the efficacy result, but they then need to analyze all the safety data and write it up and present it to a regulatory agency. Companies are very sensitive to that. You may have seen within, I think yesterday, the major vaccine companies made a pledge that they're going to make sure that none of their vaccines is released until it is deemed safe and effective enough. They're not going to be rushed into releasing something until they really understand that it's safe and effective from looking at all the data and writing it up. And when I say writing it up, under ordinary circumstances, when a company gets all these data in a large phase 3 trial of this many people, it can take 4 or 5 months to write it all up. Now, cases like this, they'll find a way to write it much faster, but it does take time. And then it has to go to a regulatory agency, and a regulatory agency, if it's doing its job, is going to scrutinize all of the data and not just simply believe what they see in Yahoo News. They're going to look at everything and be satisfied that there is a good safety profile and the efficacy is what it appears to be, and make sure that the right quality is done, the right documentation is done. So from all of that, it seems as though it's going to be some amount of time until a vaccine can be released. That first, that all the data goes into a regulatory agency, and secondly, that the agency has a time to review it all. And keep in mind, if there are several large-scale trials, it's the same people in the regulatory agencies are looking at the results of all trials. If all 4 trials ended on the same day and they got all 4 packages of data on the same day, they somehow have to review 4 huge packages of data, and it's the same people doing the same work. So I know I'm not answering the question, but I'm giving some skepticism that a vaccine will be approved for use very quickly. It can be done under political mandate, particularly if enough doses are manufactured, but it runs the risk that it might not be the right profile of vaccine. We do not know enough about its safety and efficacy profile, and that's not good, and manufacturer doesn't want it. And furthermore, if you release a vaccine that doesn't work as well as people thought, or it's not as safe as people thought, people will grow sour on vaccines in general. And that's a big problem. I'm not going to say it's a bigger problem. COVID's a big problem, but we have right now worldwide, there are probably 3 million deaths prevented per year by vaccine preventable disease. There are another 3 or 4 million more that could be prevented if vaccines were used more widely, and maybe another 2 million from other vaccines in development that one day can prevent diseases. If people lose faith in vaccines and don't use them, you're going to have literally millions or more young kids in particular who will die because they're not getting vaccines because people lose confidence. And people aren't vaccinating their kids. Other diseases are going to come back in spades or in aces of spades. We've already seen, just because of COVID, people are not vaccinating their kids as much, and we're seeing upticks in disease. So that's the ramification of getting a vaccine out too early. And yet you don't want to wait too long because people want to have a vaccine, so they're protected. So this is a very difficult debate. And all I can say is that you need to have a debate among healthcare workers, politicians, public advocates, and citizens who are knowledgeable in technical specifics, medically knowledgeable, politically knowledgeable. And everything we've discussed has to be a debate of what is the right balance of safety and efficacy, of full deliberation by an agency versus rushing and giving emergency authorization. I don't have the wherewithal myself to speculate on what is the right balance, but it's going to take some middle ground of all those factors, which is not going to happen, I think, a few weeks from now and maybe a few months from now. We'll wait and see. And that also assumes that the current vaccines will have a good rate of efficacy, which we all hope, but we don't know that either. So those are all the difficult issues that have to be overcome and understood. It's really very much a medical ethical debate of fast, get something that'll help some people, versus slow and make it better, be more certain of what you have. And how do you balance these needs? It's very difficult. And rest assured, if it's done too quickly, there'll be criticism. If it's done too slowly, there'll be criticism. If the vaccine works less well than expected, there'll be criticism. If there are safety issues that come up in 1 million people that didn't show up in 10,000 people, there'll be criticism. It's very, very hard in a situation like this to get it right. And I only hope that the blend of public health people, vaccine companies, politicians, advocates, and citizens can find the right blend, and realizing we're doing the best we can as a society to get this done in the right way. And it is hard. I'll also mention as another issue, there's rollout of vaccine from manufacturing. If let's say there's use authorization, I'm just going to throw out a date just for fun, in March of 2021, how much vaccine can be available? These vaccine facilities had to be built and fitted with equipment, people trained to work in these facilities, and maybe there's capacity of 102 million a year after it's functional. I don't know today how many vaccine facilities are functional. So on the day the vaccine is approved, I don't know whether it'll be 100 million doses, 500 million doses. It's really unknown. All I can say is the longer it'll take to approve, the more doses are available. But you'll have multiple countries, and many countries want the vaccine. There's not going to be enough vaccine to go to all countries. There's also another rather insidious thing that can happen, but it's true. If vaccine is being made in a given country, I'm just going to take a country arbitrarily like the UK, and they have a number of doses there, I would assume that the Prime Minister of the UK will say, great, you have all those doses. You may not export those doses until the day after you have vaccinated the UK population. Because I, the Prime Minister, am responsible, not to the world, I'm responsible for this country. So I embargo any export until our population is vaccinated. Something like that could happen. And that's the same sort of national embargo that can happen with food, with medical supplies, where a country will make sure that it is all set before it then thinks it's going to supply the world. Then in terms of risk groups that could be affected, I think there are 3 groups that are the highest priority. I mean, to me, without question, the first group of people who should get vaccinated are healthcare workers, the ones who are going to be dealing with people who could have COVID infection. Because they're the ones who have to be immunized. If they're not immunized and they get infected, you have no healthcare system. And we've seen the number of healthcare workers in Israel who've been in Bidud, who've been severely ill, and they're taken out. And as a result, our healthcare system is not fully staffed. Then you have individuals who have underlying medical conditions, whether it's immune related or otherwise, where they are recognized to be at higher risk of COVID. And then you have people over certain ages, and you probably do it in strata, maybe ages 75 and up, and then when you have more vaccines, 65 and up, then 55 and up. So you do it in the order of priority of those who are most vulnerable to vaccinated healthcare workers. And then through all this vaccination, if it's done enough and enough people are immunized, you get herd immunity. For many vaccines, you need an 80% immunized population, or either immunized or already naturally exposed. Whether that's the right number for COVID remains to be seen, but it's something well over 50% until you reach a point where the virus gets a dead end. You know, you have people who are infected, but enough people surrounding that person who are immune that the virus is at a dead end, and that's herd immunity. Other things you have to look at are booster doses, which might be every year. Alternatively, can you immunize with one vaccine, boost a year later with another vaccine? Again, it's unknown. Now you have the other big issue, which is vaccine refusal. And you've seen in many areas where have said they don't want to take the vaccine. There have been polls in the US saying only 50% of the population is willing to take it. There are some people who refuse all vaccines for religious or personal reasons, or they just don't think vaccines should be used, or they don't like foreign things injected into people, or they know someone who's had a severe vaccine-related injury, even at the rate of 1 in 100,000. They don't believe in control. They don't trust governments and agencies who make recommendations. They don't trust companies who are making profit from this. They believe in freedom of personal choice, not that that no one can tell them what to do. All these things blended together. There are probably 10 to 20% of the American population, maybe Israel population, who either don't vaccinate or are under-vaccinated. And more than those with COVID, they want to wait to see if a vaccine's been around and safe for some period of time before they're willing to be vaccinated. And the extent to which this factor, this vaccine hesitation or refusal is the case, it'll be that much slower until herd immunity is achieved. So educating people about the vaccine and helping to sell, and I say sell in quotes, to sell the concept of vaccination to people is going to be very important, so they have confidence to take the vaccine, and then we achieve herd immunity sooner, and then we're all much better off. So as I say, all of these will have an effect on confidence in this vaccine, the balance of safety and efficacy, and as importantly, confidence in other vaccines, and confidence in governments and medical boards who can make recommendations which balance these difficult safety and efficacy issues. Too soon, not too soon, safe enough, not too safe, and how do you balance all of that? This is going to be a huge, huge challenge for public health. And we live in very interesting times, and let's hope that we as a society achieve the right balance of all these issues, that we can get good vaccines out over the coming years that people have confidence in taking and that can help achieve a herd immunity. So I'm going to stop there and now take additional questions that you have.
Ron, look at your chat.
No, I'm looking at the last question was, is a question on flu vaccine, that it's going to be given soon. Will it affect receptivity to COVID? It's probably not going to have an effect on COVID in the sense that you may be able to take the two vaccines about the same time. It'll be tested. There's no reason a priori to think that vaccination with one will affect vaccination with another. So I say that remains to.
Ron, could you comment about the supposed protective effect of vitamin D, and also how chloroquine and remdesivir were supposed to work?
Vitamin D is probably acting by helping the immune system to be stronger. It's known to be an immune fortifier. And there are many other vitamins that will do that as well. You know, people take multivitamin pills. That's probably a good thing in general for many aspects of health. And vitamin D, more than some other vitamins, may be helpful in immune function. Chloroquine has never been proven to work, to my knowledge. There's no controlled clinical trial that shows it definitively, and to my knowledge, any recommendation of its use has been abandoned. There's an antiviral drug on the market in the US that has been used. It's made by Amgen, made by Gilead, and this works as an antiviral drug, and this shortens hospital stays by an average of about 3 days. But it's a very expensive drug given only on hospitalization, and would be given only to people who are severely ill. It wouldn't be given in general. There are many companies who are working more on routine antivirals like there are for HIV and for hepatitis C, but more antivirals that can be given as pills for routine use, that's still, I'll say, years away. It's going to take some period of time until those become available. Looking at some of the other questions, there's questions on recommendations for Professor Gamzu. Again, I don't want to get into politics, and who am I to recommend anything to Professor Gamzu, who is a brilliant, fantastic person. I guess all I'd say is I hope that he can help to orchestrate the debate in Israel that I was talking about, which is too soon, wait more, what's the right level of safety, convincing enough people to take it, even if there's uncertainty, and even if people are anti-vaccine. And if he plays that role once vaccines become available, I think that'll be very, very useful. But it'll go far beyond him. It's a job that not just one person, as brilliant and as hardworking as he is, that's a job for many people to do. Israeli companies, serious players. There are 2 companies that are involved. I'm actually a consultant and CTO for one of them, and one of the things I do on the one of the things I'm doing on the side, which is a vaccine that's coming out of the Migal Institute in Kiryat Shmona. There's also a vaccine being made by the Israel Biologic Institute in Ness Ziona, and the one in Ness Ziona should be in clinical trials within a couple of months. The one from Kiryat Shmona a bit after that. So Israeli companies are very active, and there are clinical trials, I think, already ongoing in Israel for COVID vaccine, and more will come. Flu vaccine is not expected to suppress the immune system, so it probably will not create a problem for COVID. In fact, I'd say it's actually, I'll say it's in a very funny way, that clinical flu, a flu infection, actually in a way might help the immune system to create some non-specific immunity against COVID, because your body creates non-specific immunity whenever there is a virus or bacterial infection. If you get these kinds of viral infections, it might actually help you to fight another infection like COVID, not specifically, but in terms of general immune activation. But there's nothing known about flu vaccine that should have any positive effect or definite negative effect on COVID. No doubt it will be tested, but there's no reason to think that'll be the case. There are very few vaccines that interfere with one another. There are many vaccines given simultaneously. Shots are given to little children of 10 or more different vaccine components at a time, and they respond very well to those. There are pneumococcal vaccines with up to 23 components, again, good responses. So this kind of interference is not expected to be the case. Those are all the chat questions I see. I'll take some verbal questions also if there are no more chat questions.
That's it.
Any other questions that anybody has about the ethical debate and vaccine acceptance and how to balance this soon enough, not too soon dilemma?
Okay. I don't know if you can hear me. Can you hear me?
Yes, I hear you.
Okay, good, because my screen is in a non-responsive mode, so I have limited capabilities on the screen. I just want to express our thanks and the thanks of the over 100 people who are online tonight who had the opportunity to hear your views of these various aspects of the vaccine program. I think for most of us it was an eye-opener to hear somebody speak who doesn't have the political issues to deal with and is speaking purely from the scientific side. So we appreciate the fact that you made the time, and I'm sure that I speak for everybody online that we're grateful to you for this presentation tonight. So thank you very much, and thanks to all of you. For those of you that are on our mailing list, you'll get a video link to this recording, the recording of this presentation. If you're not on our Ohel Nechama mailing list and you find me somewhere, I'll be happy to provide that to you as well. It also goes up on our YouTube site. So thanks to all of you for participating, and Ron, again, indebted to you for taking this hour and 15 minutes tonight and sharing your wisdom with us. So thank you very much.
Okay, thank you all.
Thank you. Very interesting.
Oh, you did? When?